Hello dear students, we are discussing the digestion and absorption of the nutrients in the GAT. So in the previous video we have discussed about the carbohydrates. So there we have discussed like why bigger molecules has to be broken down into smaller molecules and all those things. In this topic, so we are going to keep it simple because most of the general discussion is done there itself. So now we are going to discuss today how the proteins are getting digested and reabsorbed into the body. So let's go to this topic. So the protein digestion, the site of action primarily happens in the stomach and in the pancreas. So the primary site of action happens in the stomach itself and then the pancreas. In the stomach, what happens is the chief cells in the GI starting itself, we saw there are some cells which is called as chief cells. They will secrete or produce a substance that is called as pepsinogen. This pepsinogen will further get converted into pepsin. So this is one important thing. And this pepsin activation happens because of the HCl which is produced by the parietal cells. So we have one important substance that is nothing but the pepsin. Coming to the pancreas will produce a lots of enzymes. It will produce a lot of enzymes. The pancreatic enzymes, we can say exocrine part of the pancreas will produce a lot of enzymes, and all of them are helpful in the protein digestions. They are going to break down every single components of a protein till the end products are formed. So they have the most powerful enzymes. In the carbohydrate digestion also we saw the pancreatic amylase has the most powerful enzyme. So coming to this pancreatic juice will produce most of the substances in an inactive form. So that is the trypsinogen, chymotrypsinogen, proelastase, procarboxypeptidase A and procarboxypeptidase B. These are all called as inactive forms. All of them are inactive. Why they are inactive is because had they get activated in the pancreas itself, then this trypsin is like a severe destruction. So it can even destroy the pancreas also. The pancreas is prevented from digestion from these strong enzymes because they are kept in an inactivated state. So how do they get activated? So once these enzymes come into the lumen of the GAT, the trypsinogen, which was an inactive form, that will get activated by an enzyme. This enzyme is very important, the enterokinase. As the name indicates, it is entero. So the intestinal lumen is going to produce this enterokinase. So before that, the trypsinogen will not be getting activated. So this is produced from the intestinal lumen. See the body's mechanism, how it is trying to prevent it, but at the same time activate it when it is required. So the enterokinase will activate the trypsin. Once trypsin gets activated, the trypsin can further activate all the enzymes. It can activate chymotrypsinogen to chymotrypsin, proelastase to elastase, carboxypeptidase A to carboxypeptidase, the pro version of carboxypeptidase B to the carboxypeptidase B. So these are all the active agents we need for the destruction of the protein or the digestion of the protein, we can say. So the protein, it is primarily started its digestion in the stomach itself with the help of pepsin from the chief cells. They will form proteases, peptones and polypeptides. So these are the products they can form, but they cannot form very small molecules. That is done by the pancreatic secretions. So all these proteases, peptones and polypeptides, they will be further broken down into polypeptides and amino acids. And finally, there is another enzyme which is peptidases. They will act upon, and all these polypeptides will be broken down into di and tripeptides. Polypeptides means it has multiple chains. So they will be broken down into further dipeptides, tripeptides and the amino acids themselves. Not everything will be broken down into an amino acid form in the lumen itself. This dipeptides and tripeptides further they has to be broken down. So what will happen? It happens after the reabsorption is taking place. So these are the end products of digestion. So after digestion, the lumen has to take through the luminal side and send it to the basolateral side into the blood, just like we saw it in the carbohydrate digestion. So the transporters we have to understand. So the primary transporter here is sodium amino acid co-transporter, because both of them are going to go to the co-transporter. So both of them are going to be transported together. And obviously they are secondary active transport. So they need energy from the primary active transport, which is given by the sodium potassium ATPase pump. This pump is a huge donor of energy. Wherever you see many places, either in the GAT or in the renal system, you will always find the sodium potassium ATPase providing energy. So this is the journey of the amino acids. So the amino acid, through the basolateral amino acids, they will reach the blood vessel. They have reached it. But coming to this dipeptides and tripeptides, they are transported with a transporter that is called as PEPT1 transporter. So it's very easy to remember peptides. So the transporter name is PEPT1. Here it is not driven through the gradient of sodium. It is driven with the help of the gradient that is the hydrogen. So it is driven by hydrogen peptide to the inside. Now this di and tripeptides, they are not yet broken into amino acids. Here this will happen in the luminal cell with the help of cytoplasmic enzymes, wherein they will digest and they will form the amino acid. Again, they will be transported through the basolateral amino acid transporters. So that's how the reabsorption of proteins happen. Just remember two transporters, sodium amino acid transporter and we have the PEP transporter. Majority of the time, what happens is, even in disease, the patient generally does not land up in complete amino acid deficiency because either one of them are working. For example, in Hartnupp's disease, the clinical aspect, the Hartnupp's disease, here the primary amino acid transporters are might be defective, but here some amino acid transporters are defective. But again, we have other transporters which compensates for it. So other transporters, of course, this patient will have some amount of protein deficiency, but it is not that severe because other transporters still compensates. And the name of the disease is nothing but Hartnupp's disease. So that's why we generally don't see patients with some kind of absorption problem who is landing up with severe protein or amino acid deficiency. They will land up, but majority of the time, because of the major other transporters also, it is helping the patient for their survival. So this is all about the protein transport and reabsorption. In the next video, we'll be discussing about the fats. So we'll do it in a separate video. Thank you for watching the video. Stay tuned for more such video. Please do like and subscribe. You can put your doubts in the comment section. I'll be happy to help. Thank you so much.